drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode.
Thanks so much for having me.
Can you just start off by telling everyone a little bit about who you are and what you do?
Sure. I'm a population health researcher. I work at Mass General Hospital, also with appointments at Harvard Med School and the Broad Institute at MIT and Harvard. My work broadly is looking in large human population scale data sets to try to understand aging, both from a sort of the perspective of what's the biology of aging, but what are the different environmental drivers of aging as well, and also how that leads into many different age-related diseases.
And an important distinction that people often make in the aging field, and that we're going to talk about today in different ways, is chronological versus biological age. Can you give people just a basic sense for what those things mean and how people in this field have thought about those concepts and measured those concepts recently?
Of course. Chronological age is easy. It's just your birthday. When were you born? Let's count in years how old you are. It's a great predictor of many different age-related diseases and major common diseases, but it's imperfect because two different people can have the exact same chronological age. They can be 60 years old, but they can be very different from each other and have very different risk for disease. Mm-hmm. So some time ago now, this idea of biological age has come out, which was meant to be more of a precise way to estimate somebody's age on a biological level. So what's your level of biological function compared to someone of your given age? Is it faster, slower? Are there more comorbid conditions happening? Or are you in great health? There have been a number of different ways that people have tried to measure those over the years, but the basic concept is this, is that age is a great predictor and a great starting place for understanding your risk for tons of different diseases, but it's imperfect and there's a lot of information lost. And the thinking is if we can go into the biology instead, that will really give us more nuance and more precision, even among two people of the same age. What state are you at now? What's your future health trajectory look like? Where have you been?
Mm-hmm. And can you give us a sense for so when these two numbers start to deviate, it's really easy to think about it if you're doing things that are incredibly unhealthy. So it's very natural for people to think, and I'm sure this is borne out in the data, if you start smoking cigarettes at an early age and you're smoking 3 packs a day for decades, your biological and your chronological age are probably going to deviate. You're probably going to be biologically older than you otherwise would be from just knowing your calendar age or your chronological age. Can you give us a sense for how big some of those effects are and whether or not when these two numbers deviate, for example, is it always that doing unhealthy things makes you age faster, or can you also do things that move you in the other direction?
Well, this brings up an interesting dilemma because there's what's true and then there's what you see in epidemiological data, and those two things aren't always the same. I'll give you a classic example. For decades, the epidemiological data tend to show this kind of J shape in the association between intake of alcohol and mortality risk. So starting from no intake whatsoever, you intake a little bit, your mortality risk starts to go down, and then you intake more and more and more, and then it starts to go up.
I see. So this would be the classic, very widespread notion, at least in my experience, that if you drink a lot, that's bad. But if you have one or two drinks a night, that might actually be better than nothing.
Right. And it turns out, from what we know now, from more sort of advanced statistical modeling we've been able to do and much better ascertainment of that phenotype in the context of these epidemiological cohorts, turns out that that's not true. It's really easy to see it in terms of statistical association tests. But it turns out if you're actually able to model it the right way, and there are a lot of really tricky ways why you tend to see that. For example, if you compare it against any never drinkers, but some of those never drinkers are former drinkers, most people stop drinking because they have some sort of health condition. Their doctors tell them to stop, right? So there's all kinds of these reasons. Turns out, if you're able to actually model it correctly, the safest amount of alcohol to drink is 0. So if we sort of sidestep the problem that there's going to be a lot in the epidemiological literature that might tell you what's healthy and what will put you in one direction or another versus what's true, it should be, in theory, the case that, disease and age-related diseases and aging more broadly, you can think of it kind of as like an accumulation of damage on a biological level, an accumulation of deficits, and this is why it leads to disease. So it should be the things that we know are bad for health. Smoking is a great example. Alcohol intake is a great example. Lack of physical activity. They are associated with so many different diseases long term. And the reason why I think they go to so many is they probably have some impact more centrally on these kind of aging mechanisms that touch all of these different diseases simultaneously. The problem is right now, where we are in the state of, I would say, convincing large-scale human data on what moves these aging clocks is not well known. I think there's a lot of research in mice and animals and worms, and there's some small-scale human studies. We're trying to do some work now going deeper into the different things in your environment, behavior, lifestyles, even interventions that are changing these on the sort of large human population scale. But I think beyond a couple really basic things, like I said, smoking, alcohol, physical activity, the extent to which this or that is surefire going to improve your biological aging or lead to some kind of detriment in biological aging, I wouldn't say we have the perfect catalog yet.
I know this isn't going to be the focus today, but you mentioned this interesting pattern that's been observed in the epidemiological literature with respect to alcohol that turns out not to be true. And I think this is important, an important general thing, because, in the field of epidemiology, broadly speaking, when we're looking at associations that have to do with human health, these are intrinsically interesting areas of study because people are all interested in human health to some extent. They tend to get a lot of attention in the media, and yet, as I think you probably know, a lot of those studies could be very, very misleading. And you just gave us the alcohol example. When you see results in the epidemiology literature, how do you start to think about, or how can people think about weighting the evidence that we see in some of those epidemiology studies? So for example, when you mentioned that alcohol result, and this is sort of how I do it, I guess, is one thing I like to do is I like to ask myself, what's the result that people want to be true? So in the case of alcohol, right, it would be very convenient if having a few drinks a week was healthy for you. So we have to sort of discount that because we're all biased in that direction, most of us at least. I would love if it was true if you had a couple drinks, every day, that that was the healthiest thing you could possibly do. And then two, there's this question, I think, of biological plausibility or first principles thinking that I think people often don't do when they look at these associations. And based on how I understand alcohol metabolism, it's very difficult for me to think about what is biologically healthy about ingesting ethanol or its metabolites and how that could possibly help you live longer. How do you think about evaluating those types of association studies?
So we could think about it in terms of a couple pillars that at least I think about, and I think our group more broadly tends to think about on terms of what is a convincing amount of evidence that we think it's plausible or interesting or far-fetched, or we're just not sure. I will say that the focus of a lot of the research that I do and that my colleagues do in our group, it's focused on the power of large-scale human data. So there are other people that use other kinds of animal models, things like that I mentioned, but for me, the first pillar is, have we established in a large human population, and have we replicated it in independent and different populations from the one in which you started? For me, that's number one. You need to be able to demonstrate that what you see is generalizable and consistent across different ways you slice up the population. So we want a large human population. We want one that is sufficiently generalizable to you and me and everybody else. If we were to have a large population of 500,000 people, but every single person in that cohort has Alzheimer's disease, it doesn't really tell you a lot about how something might work and the dynamics of it within the broader population. So number one, is it a large enough population that is representative and something can be generalizable, and can we reproduce that effect again and again from this population to another one to another one? The second pillar we can think about is, how did you measure what you're trying to assess? The classic example everyone loves to pick on is it a self-report questionnaire, or are you getting your assessment from some kind of biological data?
Yeah, this
in
is
general,
one of my,
but not.
this
Sorry,
is one
go
of my
ahead.
pet peeves, the self-reported data.
Well, I mean, and speaking of alcohol and diet, this is one of the most difficult things to assess in terms of self-report. And it's interesting, some kinds of self-report measures are more reliable than others. Alcohol is a notoriously unreliable one, whereas smoking status, people seem to lie about that less for whatever reason. But this is a classic example where if you were to take a questionnaire item versus looking at the biology, you can get much different stories, and one gives you a more reliable output. So let's stick with the same alcohol example. Like I said, if you look at questionnaire data, you'll see the kind of incorrect association until you can start to realize there's some confounding within the way you've constructed that measure, and we can start to tease that out. So nowadays, people will exclude all of the previous drinkers, and they might exclude some of the never drinkers and just go among drinkers, smallest to the lowest. So there's just a preponderance of studies showing protective effect of some small amount of alcohol. But the biggest studies that are coming out in Lancet, these huge global meta-analyses, these huge global efforts, the safest amount is 0. But we can look at a different set of evidence, which instead of self-report information might use genetics. So there's this concept of Mendelian randomization, where you take a genetic variant that you use as a proxy variable for your exposure of interest. In this case, alcohol. There are known genetic variants that will associate or correlate with alcohol intake. So we can use the genetic variant, which has the benefit and the advantage of being randomly assigned at birth. So we're removing some of the confounding in a sort of patterned way that exposure could be meted out across the population. We can use that instead as a proxy and measure what's the association of that with the outcome of interest. And when you use the genetic proxies, the genetic variants in a Mendelian randomization study, it's much clearer. The safest amount of intake is 0, and then as you go up in exposure, it just increases your risk of cardiovascular disease and other things. So that's number 2. How are we measuring what we want to measure as the exposure of interest? There are, I would say, self-report, you've got biological proxies, you've also got randomized trials and interventions where you at least are able to control a little bit more of the confounding and the bias and the way that things are patterned among people. So that's really number 2. The third really comes down to, I would say, how can we evaluate these across populations? So, and this kind of ties back into the first example as well. In my opinion, it's not just something that we in one study should be able to show has had some effect, even if we validate it in external populations a couple times. Is the field as a consensus coming across this? Is everybody with different study designs, there are different ways of computing it in models pointing at some kind of similar direction of effect and magnitude effect? Or is it that only this group of researchers and in this one study with this one particular model type found something, And then turns out no one else can replicate it. We've tried it in other cohorts, it didn't work. We might have tried it in the same cohort with other model types, it didn't work. So I think some things are more flimsy. And the other thing that we've done, especially in the genetics community, to try to increase our confidence in what we find is by going large scale. So the easiest way to think about this, again, in the context of genetics research, is it used to be you wanted to know the association between genetics and some outcome, let's say cardiovascular disease. Well, then you might hypothesize this one gene for some biological region might be associated. You'll do a statistical test. We'll use a p-value of 0.05, and it might just skirt through. And then we'll say, okay, that looks great. I think we found it. Publish it in a journal. That's not really the way we do it in genetics anymore. Now we'll do genome-wide association studies where we'll measure all of the genetic variants we can, or at least usually all of the common genetic variants we can. And so now you have just tons and tons of tests. And so the threshold for what becomes statistically significant becomes, in terms of a p-value, much smaller. You can think about it, the threshold becomes much, much more difficult to prove something is associated with whatever you're trying to look at above and beyond everything else. And what that's done is just completely changed the number of false positives that we've had in genetics research, for example. There used to be a huge preponderance of false positives, and now that's been dramatically slashed by the scale at which we do this kind of research. You know, we're trying to find the one signal that's bigger than all of the other signals that exist, instead of we're just looking at one and does it skirt by in terms of some borderline statistical significance. So again, I'm a little biased in this direction. Like I said at the outset, our approach for everything is large-scale human data and across diverse global populations, because we think that's what gives you what's common in biology across groups in the world, and not just what's noise.
Right. And that's I'm not going to say that's everything that we care about, but most of the things that people care about are things we have in common. Like if people care about longevity, pretty much everyone cares about that. So there should be some common core factors that are true across the board, across all human populations.
It should, and it's not just for aging. I mean, think about it for anything. Think about trying to develop drugs or develop pharmaceuticals, right? If you want to develop, even for covering your own bases economically, if you're a pharma company, you want to develop something that works for everybody and not just in a couple of people. Then you bring it to market and you find out, oh, shoot, you know, it only works in this one specific group. It's not working for anybody else. So I think no matter what we're aiming for, it's what's common across people, and not, you know, what's just noise in one particular setting.
So aging, you know, again, inherently interesting topic. Lots, you know, everyone's limited by their own mortality. So we you know, we all want to live long, healthy lives, at least. So Healthspan, I guess, as opposed to just raw lifespan. What's becoming really popular are these aging clocks. There's lots of commercial examples of this now. You can buy these, you know, different aging clocks, and they tell you your chronological age compared to your biological age. So basically, are you younger than your calendar age would suggest, so to speak, or are you older? Do you have maybe some work to do to get healthier? Can you give us a basic sense for how most of these clocks work today? What sort of biological markers are they based on? Do they work well, and how well do they work?
The idea of a clock has evolved over the years. It started using DNA methylation information. I mean, these came out about 10 years ago or so. The first one was Steve Horvath's clock, came out around 2013. But the idea is basically, can you take some kind of biological information and can we use it to predict somebody's age?
Yep.
The first insight was that DNA methylation as a sort of biological information tends to associate or correlate very closely, at least some CpGs, with calendar age, and we can develop a very predictive model of age. So what usually happens is people will take some kind of biological information. Again, the first case, it was DNA methylation, put it in a model, and then have the output you're trying to predict be chronological age. You can then make a nice plot of, you know, what's your methylation predicted age versus what's your chronological age. You can calculate the correlation between those two. That will give you your, so to speak, accuracy of these kinds of clocks. And then the idea being that there is this kind of, like you mentioned, this divergence between what your biologically predicted age might be and your actual calendar age is, and then that will be informative for, telling you about future risk of all kinds of different age-related outcomes and phenotypes. The problem is we've had to go through many different iterations of biological clocks over the years because they haven't always been as predictive of the actual aging phenotypes we care about as we thought they might be. So in thinking about methylation clocks, and then I'll get into other omics because I think probably other omics are more important for this ultimately. But methylation clocks, the history was such that you would develop these models, which were usually linear models, things like lasso models or things like that. And they would be really great at predicting chronological age. An accuracy of, in the high 90s. The problem is, sorry, the problem is, sorry about that.
No worries, no worries. I was in the same boat the last few days.
I'll start from the beginning of the sentence.
Yeah, no worries. Yeah, so most of the clocks so far that are out there that have been used that people will see commercial examples of, most of those today are based on DNA methylation.
Yeah, that's right. And the problem is those initial DNA methylation clocks, they're great at telling you about your chronological age, predicting what your age is, but they're kind of lousy at telling you about your future risk or telling you about your risk for morbidity, clinical information, or mortality.
Can you actually, before we go further, why is there a good reason that DNA methylation tracks with age or predicts age? Does our DNA tend to become hypermethylated over time or hypomethylated over time?
There are different theories. Someone who's written a lot about this in a very intriguing way is David Sinclair at Harvard. He has his entire information theory of aging, he calls it, which is largely boils down to the fact that aging entails basically a loss of epigenetic information over time. So DNA methylation, which are these chemical marks on top of DNA, aren't static across life, and they're certainly not static or consistent across cells in your body. And so what they are is very dynamic, and he has some compelling research showing that, at least in some of the studies that he's worked on, some of these epigenetic regulatory mechanisms sort of will have dual or shared roles where they might be involved both in epigenetic regulation, but then also DNA damage repair and somatic maintenance and things like that. And as time, when they have to move back and forth between these roles, they get lost and you tend to lose information. So the theory is that epigenetic regulators over time will change in some kind of stable way in terms of thinking about them through time, and that tracks really well with age. So in some cases for methylation, the analogy of a clock is apt because it just seems to kind of tick along with time. And so that's why it's so good at capturing chronological age information. The problem is that there are so many steps that have to go from DNA methylation to the end result, which would actually influence a phenotype we care about, which is basically changing protein levels, that those methylation marks don't always correlate strongly with ultimately the protein, either for the gene that methylation mark is in or something more downstream or distal, that there tends to be this disconnect. And this is why, or this is at least our theory, why these methylation clocks are great at basically counting the ticking of time, but might not necessarily be so well associated with the actual aging phenotypes that we care about, whether that's frailty or age-related disease or ultimately mortality risk. And this is why there have been several sort of generations of DNA methylation clocks. They left these first generation, as they're called now, clocks pretty early and moved on to so-called second generation clocks. So the first generation clocks would be like Horvath's clock or Hannum's clock. Second generation clocks would be things like pheno-age or Grimm age, as they're called. And these are no longer models that are built on taking DNA methylation to predict age anymore. These are models that use DNA methylation to predict some type of phenotypic aging phenotype, which would probably be constructed from either different types of blood biochemistry markers, albumin, creatinine, inflammation markers. In the case of Grimm age, they also use smoking. So things that are already telling you about some kind of morbidity or function or sort of physical functioning, and then they're using DNA methylation to capture that instead. Those are much more predictive of mortality and morbidity than the first generation clocks. And then you have what are called third generation clocks. These are things, there's one called Dunedin Pace, which is built on sort of assessing over time. So this is kind of like a longitudinal measure of DNA methylation and what that tells you about change over time in DNA methylation. But there's always this question mark, in my mind at least, alongside a lot of these methylation clocks is, are they capturing the real biology that is what's driving the aging phenotypes, or are they just capturing the passage of time? And there was an interesting paper that came out, I think this January, from Vadim Gladishev's lab at Harvard, where they built a new kind of methylation clock, but instead of just using any CpG site in the methylome, they only picked among CpG sites that they demonstrated has some causal relationship with aging phenotypes through using Mendelian randomization, which was that genetic type of genetic association testing I was talking about before. And unsurprisingly, what they find is they build a new clock based on these causal CpGs, and they find that most of the major clocks that have been established to date aren't really enriched for any of these causal CpGs. And this kind of lines up with what we know more generally about the biology and kind of what are the correlations between different stages of biology, from genes all the way to proteins. I mean, the central dogma should be you have genes and then you go into transcription, you make mRNA, then you make proteins. The reality is that across many genes, but then especially in development and aging genes, there's a pretty strong discordance between gene expression and protein expression. And the real reason why DNA methylation would matter for any type of physical functioning or disease phenotypes is that it should ultimately change gene expression. You know, methylation famously is the mark that will turn a gene on or off, right? So it should be having some effect on changing gene expression. Well, it turns out the gene expression signal for many genes throughout the genome isn't always very well correlated with the amount of that protein that ends up in your body. So
is that just because, you can turn a gene on and then that gene can be transcribed into a protein? There could be a one-to-one sort of correspondence between how much transcription you get and then how much protein gets made. But there's other cases where, due to post-transcriptional modifications, things that happen after the gene is turned on, that you get, a deviation between how much gene product is produced and how much protein ultimately gets made.
Yeah, the thinking is that a lot of what's going on here are what are called post-transcriptional or post-translational modifications. So you have a gene, you have a transcript that's made, you make an mRNA. That should then ultimately lead into translation into a protein. But we know that at different stages from gene transcript up to the protein, there are a lot of different things that can happen. And even after the protein is made. So there are post-transcriptional modifications that can change, and then that will maybe change how much of the protein is expressed. But then even after the protein's already made, there are a lot of different modifications that can happen to the protein afterwards that will change, how much of that protein you see in the body. What we found in some of our research, and this can maybe lead into some of where I think some of the most promising avenues are now in terms of proteins and proteomics, is that some of the most age-associated proteins that we tend to see in some of our work, and certainly other papers that have come out previously as well, is that proteins that tend to stick around in the body for a long time, so things like extracellular matrix proteins, for example, that have really long half-lives, these are very age-associated. But because they stick around so long, they're basically around for a long time to be subject to all kinds of degradation and damage and modifications. And so every time that happens, it's just pulling away the amount of that protein further and further out of correlation with the initial gene expression signal. And so
that makes intuitive sense to me. Like if a protein is very long lived, especially if it's outside of a cell, it's in the extracellular space, it's sort of exposed to the environment of that person's body. And so it will sort of, I would think it would naturally then just sort of capture what that person's exposing themselves to more than other proteins.
Yeah, exactly. And it's kind of what we're learning is a kind of a tenuous exercise to start by measuring something very upstream like methylation or gene expression
Hmm.
and just expecting that that's automatically going to capture the real phenotypic variants that you care about
Hmm.
for disease or frailty or anything that we care about in terms of age-related outcomes or phenotypes. And so this is why you've seen a boom in the last few years, at least, of people moving into proteomics
Yep.
to try to measure aging
Yep.
instead. I mean, proteins inherently are what we care about. They're often what are the causal drivers of disease. They're also usually the targets for therapeutic approaches and interventions to alleviate disease. So there are a lot of reasons why proteins are kind of the layer of interest, so to speak. And so now that the technology and the data are sort of becoming available to look at that directly, people are starting to look directly at that instead.
Hmm. Yeah, and that kind of brings us to some of your recent work. So basically, the idea here is, okay, we want to study aging. We're interested in this idea of aging clocks. Well, instead of looking at mRNA, let's look at something that's closer to the aging phenotypes we actually care about. Let's look at the actual effectors
Yeah.
of aging, basically, and that would be, in many cases, proteins themselves.
Yeah, exactly. And so, you know, with all of these advancements, the rate limiting step is always what data are available.
Yep.
So when the first epigenetic clock was made a little over 10 years ago, Steve Horvath spent, I think, years, if I'm getting the story right, just going out and curating and collecting all this different methylation data he could pull together and try to make a model that was big enough to build his clock. Well, these days, now we have these big human biobanks that are collecting all kinds of biological data, and now they're starting to invest in proteomics, although it's very expensive. So now we have the opportunity within the context of some very well phenotyped and characterized biobanks and cohorts to start to look directly at the proteins. And proteomics is not at the stage yet where you have, for example, methylation or transcriptomics data where, theoretically you'll have a much broader coverage of the genome. I mean, the latest DNA methylation arrays, I think, look at close to 1 million or so CpG sites across many, many genes. The best proteomics assays we have right now are in the neighborhood between 5 and 11,000 proteins, but we know there are, of course, far more in the actual human proteome.
How big is the human proteome? Can you tell us?
I couldn't tell you a number. I think it's...
Is it like hundreds of millions of proteins, probably?
Millions, I don't know.
Yeah.
But I think there would be more proteins than there are genes because you all have different kinds of isoforms and different and different kinds of proteins. So I would at least think, if you want to think about how many proteins are in their body, think about it as
20,000 genes. There's got to be quite a bit more than that.
There's going to be many proteins. So, we have okay coverage of those now, but, different proteins are different sizes. They're in the bodies at far different abundances from one another. So it becomes very tricky from a technological standpoint to capture all of them, which have, have much different abundances and different levels and to have different sensitivity across different ranges. So it's an ongoing field and it's evolving rapidly, but there have in the last 4 or 5 years there has been an explosion in terms of proteomics data that have come into the world available to researchers. The biggest explosion by far was last year. The UK Biobank made available to the world and any researcher who wants to use them proteomics data for about 50,000 people. And why this is so important is because this is in a biobank where it's not just 50,000 people with no information. It's 50,000 people where you already have whole genome sequencing, array genotyping, where you have questionnaire data, full blood biochemistry panels on everybody, and you have linked mortality and electronic health records for everybody. So if there's a disease with an ICD code, you can study it in relationship to all the biological data that's become available. And so this was the sort of foreground for some of the research that we've been doing, where again, our whole approach is try to leverage as rich, large-scale human biological and sort of phenotypic data as we can to develop tools. And so we had this idea anyways, well, what if we made a proteomic clock and we built it from this huge cohort? But because we have so much other information, we could then at the same time test it really systematically across tons of different diseases. And this has been a hypothesis around for a long time. It's usually called the geroscience hypothesis, which is that There are sort of known biological hallmarks of aging. I think now we're up to 12 that are sort of considered canonical. Things like telomere length and loss of proteostasis. Epigenetic changes is one as well.
What does loss of proteostasis mean?
Protein stability, basically.
I see.
So, but now they consider things like gut dysbiosis another hallmark of aging, DNA damage. So there was an initial hallmark paper in Cell that had an initial set of these, and then they just updated it last year to now these sort of 12 pillars or biological hallmarks of aging.
So these are things that are measurable, that reproducibly track with aging very well.
That's right. And we see them conserved across species, and we see them again and again. And the theory has been that these sort of hallmarks of aging, or aging as a biological phenomenon, is probably common to all of the major age-related diseases. And people will demonstrate that in, this study from this one hallmark to this disease, and another study, this one hallmark to another disease. And so there's been kind of a patchwork of evidence supporting that so far. Our thought was, well, if we have a large enough data set and we have electronic health record, we could probably just test that all in one go among the same people. And so that was one of the sort of motivating ideas for this work now is, well, if we can make a good enough biological signal of aging, and this geroscience hypothesis is correct, then it should be that this biological signature of aging is going to be associated with all the major age-related diseases we can measure. All we need to do is pull them out of an EHR, or electronic health record, and we can test it.
Can you tell us a little bit more about this data set you were working with? Where does it come from? What's it composed of? And how'd you get your hands on it?
So the UK Biobank is a very, very well-known biobank or kind of epidemiological cohort study, at least very well known in this sort of large-scale epidemiological community. It tracks 500,000 people in the UK who were recruited between 2006 and 2010. And they did a whole battery of initial testing on these people when they first came into the recruitment centers. So they took blood samples and they stored them so they could do future assessments later as technologies developed to do different kinds of things. So now they have...
And who were these people? Did they volunteer for this? Were they selected for some reason?
They're volunteers. The UK Biobank tried their hardest to make the population within this cohort study as representative of the UK population as possible. They didn't succeed 100%, but nobody really does.
Yeah. But it's probably quite representative as far as these things go.
It's reasonably representative. In general, they're more healthy than the UK population, and there are definite differences that can and have been observed and written sort of extensively about. So it's always a caveat when you work with these biobanks and data sets that it's not perfectly representative of the UK population, but it's a decent approximation, and it's far better in terms of, because of the scale at which it was gathered, than a much smaller data set where you're much more likely to have some kind of bias in the way it was sampled.
But so you've got a data set which is a large number of people had blood drawn, had various biomarkers measured, and you sort of have that information across time for a very large number of people.
Well, at this stage, they have a lot of the rich information comes from baseline. So between 2006 to 2010, when people were enrolled into the study, they came to an assessment center, they had blood drawn, they did a whole suite of anthropometric measurements, BMI, impedance measures for body fat, height, weight, all these kinds of things. They had a clinical interview with the trained clinical interviewer. They did questionnaires, they did blood biochemistry. So they did a lot. They did spirometry for lung function and things like this. So they did a lot of testing at the beginning. In a subset, they followed them up in terms of specific measurements. So at baseline and then also at some follow-up, they did MRI measurements and imaging studies on several tens of thousands of people. At least I don't remember the N right now. Also in several tens of thousands of people, they did accelerometer data. So they gave people Fitbits for a couple of weeks and they pulled in all those data as well. But the real sort of longitudinal data over time has been that they continue to pull in information from the linked electronic health records and mortality registers in the UK.
I see. So we've got health records over time plus a baseline set of measurements.
Right, exactly. And so, they pull these in at different times because the sort of register is a little bit different in Scotland versus Wales versus England. But in general, what they've done is they've pulled in across time. And so now they have about 10 to 15 years of follow-up information of from that time at baseline, what are all of the diseases and diagnoses that you've accumulated? We have the exact date of each one. They also go back retrospectively, so we know in the time preceding when they were recruited, what diagnoses did they have as well. And that's where the really rich longitudinal information is. It's thin on longitudinal biological information. In terms of the whole cohort, there's no real repeat assessment of anything. For genetics, of course, that doesn't matter because genes and DNA are stable across life. But for the other sort of biological layers of information that are much more dynamic, there's hope to be able to have repeat assessments at some point in time. But at least right now, they have genomics in terms of array genotyping and whole genome sequencing in the full cohort. They have metabolomics in, I believe, the full cohort now. And now they have these proteomics data in about 50,000 participants, which is far and away the largest proteomics data set that has existed to date. And so it's been, these data is, UK Biobank is for lack of a better term, a public resource. That doesn't mean that it's free, but it means that anybody and any researcher can apply for access. There are different fees for access based on different tiers of data that you would like to get your hands on. So the most basic access tier is relatively inexpensive, but if you want the most comprehensive data tier, which has proteomics data, whole genome sequencing, all these things, it'll cost you close to $10,000. So there's a pretty broad range. So I don't want to say it's public in the sense that anybody could just download it now. And you have to be a vetted researcher and you have to sign a data use agreement. They need to make sure that they're secure in terms of who is accessing the data and for what purpose. But as far as data sets go, it's relatively open and easy in terms of the process to apply and to try to get your hands on some of this for your own research.
So tell us a little bit more about the proteomics data. What kind of proteins are in this data set? Are there any significant patterns, like types of proteins that are present or absent?
So the proteomic assessment that was done so far in the UK Biobank was on a platform called Olink Explore 3072. It assays about 3,000 proteins, and that sort of larger panel is made up of sort of 4 major subgroups of panels that Olink has previously developed. And these are proteins that are largely related to oncology, neurodegeneration, inflammation, or sort of cardiometabolic phenotypes. So, it's definitely kind of a hedging your bets type of approach where they've developed these panels over time with proteins that are where there's either known information that these are biologically relevant for some of the major phenotypes we care about, or that are high probability targets for future discovery that we haven't looked at yet. So 3,000 proteins is okay. It's certainly not by any stretch of the imagination the largest panel that exists even now, and even Olink since then has a new flagship panel that covers 5,000 proteins instead of 3,000 proteins. So it's continued to develop even since these data came out about a year ago. And I should say the data came out a year ago to the public, but they came out a year before that in a sort of pre-competitive data release to all the pharma partners who financed this project. So this was kind of an interesting phenomenon, even in terms of sort of global community science, in that a consortium of about 12 pharma companies together in what was called the UK Biobank Pharma Proteomics Project came together to finance the sequencing of this proteomic panel in 50,000 participants. Most of them were selected generally from the population, but then they got to cherry pick some to be enriched for different diseases they cared about for their own sort of drug development pipelines. And in exchange, they got sort of early access to the proteomic data for a year before anybody else did. So by the time the general research public were given access to these data, the sort of big flagship papers describing these proteomics data sets, were already under review. But there was still lots of opportunity to explore the things. But I think there's lots of opportunity to go more. Again, to give you the range of how many proteins can we assay right now in terms of these population data sets, there's the flagship Olink platform, which is about 5,000 proteins. That's called Olink HT. SomaScan is another company that doesn't use antibodies to detect proteins like Olink. They use aptamers instead. They have their new flagship panel that will measure about 11,000 proteins. There's lots of really interesting proteogenomics work that's been happening in the last year or so, trying to compare the differences between these platforms. And given they have different technologies for detecting proteins, does that mean you're measuring different things? There's been a lot of interesting work coming out there. And then the other area is something like mass spectrometry. I don't see that generally as much in these large cohorts. I think there's a lot more difficulty in terms of what you find there. I mean, mass spectrometry, basically you put your samples through these, then you'll get lots of peaks, and then you have to annotate those peaks in the signal you find to, which protein is which. So some of them you can annotate and some of them you can't. So a lot of mass spectrometry research right now is trying to figure out what are we seeing. So, but you potentially have all the signals there. So that's, the pros and cons of that approach. These antibody aptamer based panels, the approach or the sort of trade-off is instead, well, you're getting just these few thousand or 11,000 proteins, but we know exactly what each of them are.
And so this proteomics data set, you've got several thousand proteins in it. What exactly is the measurement you have to work with here? So their proteins vary in their expression levels between individuals, across cell types and tissues in the body. Do you just have sort of the average expression level of each of these proteins, or what are the actual measurements here?
Yeah, these are all from plasma, and so we have plasma expression of each protein. The way that they're given in the UK Biobank is it's a relative normalization. So the proteomics data are actually given to you already normalized in a particular way that that Olink as a company sort of will normalize the data within the batches of when they generate the data. And there was actually a very extensive QC process that went into looking at these proteomics data by this sort of pharma consortium at the outset. So the data that are now available to the public are sort of these normalized post-QC proteomics data.
Okay, yeah, basically blood samples are taken, and then you got measurements of proteins in everyone's blood plasma. What are some of the basic headline results here in terms of how this protein data set tracked with biological age?
The headlines are a few. The first is that, using blood proteins, we can make a proteomic clock just as accurately as a methylation clock. But that too, so, that's known, but, proof of concept that we can use large scale proteomics data to achieve a similar thing. And I will say, we just tried a sort of first generation clock here. So just take all the blood proteins, put them in a model to predict age. Turns out, if you start from 3,000, you only need 200 to actually estimate somebody's age. And you can estimate it just as well with 200 versus all 3,000.
Yeah.
And you
that
can
makes
go
sense.
all the way
Like
down
some
to
proteins
20.
are going to, be very, very intimately involved in the aging process, basically.
Exactly. And what we even find is that you can go down to just 20 proteins, and that will still get you 95% of the accuracy of the larger model. So those 20 are very multidimensional and heterogeneous in what they are and what they do. But one of the headlines is we can capture aging from blood proteins, and you actually don't need thousands of proteins to do it. The second headline is that if we pay attention to how we develop our models, we can make a proteomics clock or a proteomics-based aging signature that's very generalizable to different populations. So we built this proteomic age clock in this cohort, the UK Biobank, in about 45,000 people. And then we tested how well that model works in two completely different data sets that had the same proteomics sort of panel assessed. One was a cohort in China called the China Kadoorie Biobank. We had about 4,000 people there. The other was a cohort called FinGen, which is another similar biobank study in Finland. Those participants are very different from the UK Biobank in terms of their age ranges, in terms of their morbidity profiles. FinGen participants, for example, everybody who has proteomics so far, they're all healthy. Mortality is around 1% or something like that. Very few people have illnesses. The China cohort, on the other hand, fits kind of it's a case cohort design for ischemic heart disease. So what that means is among the people who they did this first set of proteomics assessments, half of them have known ischemic heart disease, and the rest are just a random representative sample of the cohort. Of course, both of those populations are very different in terms of geography and genetic ancestry than the UK biobank population as well. And so that really wasn't a given that this would work so well. And it's also, unfortunately, not something that's usually done in the context of these sort of biological aging papers that have come out previously, and certainly not in terms of clocks, where there's been an emphasis on sort of rigorous validation of models across ancestry groups and across global populations. But if any of this, again, like we were speaking about before, if any of this is going to matter to telling us something about aging biology that's common across people that we can leverage for preventative health or developing therapeutics or anything, it's got to be common across everybody, right? And so that was number two. And that involved a number of interesting developments that we had to come up with in terms of how we train these models and how we renormalize data and things like that. And we can certainly go into those details if you want. The last thing that we find is, again, the headline is that we end up being able to support this geroscience hypothesis, which is that we can train and develop this aging biology signature based on blood proteins. And it is associated with more or less all of the major age-related chronic diseases that you can study in the UK Biobank, where you have enough people who have developed those diseases over the amount of follow-up there is so far. So that's mortality plus 18 different major diseases, things like dementia, Alzheimer's, chronic kidney disease, chronic liver disease, heart disease, stroke. So the number of diseases that we're able to associate with this single blood measure of aging is, as far as we understand, unprecedented. And it's just the power of the data set that were made available to us. Something of this scale hasn't really existed before, and what it affords you is your ability to look systematically, comprehensively in this way. We also did end up finding that this blood proteomic signature is strongly associated with a number of other aging phenotypes, so things like physical frailty, hand grip strength, lung function, blood pressure, BMI, also some of the canonical blood biochemistry signatures of aging, things like IGF-1, which is a growth factor, things like telomere length, which is one of the other cellular hallmarks of aging, C-reactive protein, which is an inflammation marker. And we can measure, and they will associate with all these things, even among the participants, the sort of subset of participants who have no major chronic disease or no major disease diagnosis to date. What that means is it's a signature that tells you about future risk of disease, but it's something that we can capture meaningful variability in, even among people who haven't developed any of them yet. And it will still associate with all of these sort of intermediate phenotypes. Cognitive function is another one. So, it will tell you you're on that pathway, and we know that it's sort of associated with the functional things that are happening from in between, you being young to, ultimately having a full-blown, major chronic disease or even multiple at the same time.
Can you give us a sense for how, give us a sense for some of the distributions here. So if you take a slice of people of a given age, I'm getting close to 40. Let's say you take all the 40-year-olds in the data set. How much variance is there in the deviation between chronological and biological age? So for example, if you take my blood or you take, a bunch of people's blood and you do this type of work, can you find a 40-year-old who biologically looks like they're 30? Can you find one that looks like they're 50? Or is everyone kind of within a few years of the chronological age? What's the spread like?
There are outliers who are extreme cases like you're describing. It's unclear to us if they're noise or that's real. But if we look at more the broader population distributions, it's plus or minus 6 to 10 years. So when in our paper, when we look at the associations, we look at sort of what's the association between your proteomic age clock and the disease per 1 year change in proteomic aging. The phenotype we use is called a proteomic age gap. Other people call it acceleration or deceleration. All it is you take your protein predicted age and you subtract your chronological age from it. So are you plus 5 years, plus 10 years, minus 5 years, et cetera. But to give you a scale in terms of the effect across that distribution, If you were in the top 5% of this proteomic age gap versus the bottom 5%, which is about, gosh, I don't remember off the top of my head, it's about a 10 to 20 year jump across those two extremes, then we're talking about a 5 to 6x increase in your Alzheimer's risk in the next 10 to 15 years.
I see. Okay.
So if you're at the extremes of these distributions, your difference in risk for developing a number of these diseases across
could be considerably high or considerably lower.
Exactly. And that's what we also find among those who are in the lowest sort of parts of the distribution. Again, so let's stick with dementia and Alzheimer's. If you're in the bottom 10% of this score, so you're going to have a negative number, you're going to be somewhere between 6 to 10 years younger in terms of blood proteins than your calendar age. Less than 1% of those people in the next 10 to 15 years, even if they're already 65 years old right now, will develop Alzheimer's or any kind of dementia. So it's really powerful at telling you who's really at high risk, but it's also really powerful at telling you who's at really low risk as well.
And how do we know anything about how malleable this biological age number is per unit time? So for example, let's say at some point in the not too distant future, you can buy a test like this and you find out, oh my God, I'm actually I would like to be aging much more slowly. Maybe I'm 60 years old. My biological age is 65. Let's just say something like that, whatever. And I'm actually, at a much higher risk for some form of dementia or what have you, above and beyond what the average 60-year-old is actually at risk for. And I want to go in the other direction. How feasible is that? How feasible is it to get my biological age back down to my chronological age of 60 or do you even dip below that? Is that feasible at that age?
That is the $1 million question. And it's the question that we get most often in response to our research, which just came out a couple of weeks ago. Okay, is this a death sentence or is there something I can do about and change it? Our suspicion is that there's something you can do about to change it. I mean, blood proteins are dynamic. We know that. We know also that, aging ultimately leads to the development of chronic diseases. And we know that you're able to change the trajectory you're on for different chronic diseases. There are effective preventative measures for cardiovascular disease, for lung diseases, all kinds of different chronic diseases. So the question that we're most interested in is, if we're able to give out some kind of intervention that will change your future trajectory for risk of these major diseases, are we going to see that reflected in the biological age signature? Then it becomes a really enticing tool in the context of preventative health and preventative medicine, because of what it can tell you is, let's do a baseline assessment. This is the trajectory you're on right now. For mortality in 18 different chronic diseases, this is what your future looks like 10 to 15 years from now. But let's course correct. Let's work with your physician. Let's try some things out, maybe physical activity, maybe it's diet, maybe there's kinds of things that you can work out with your physician. Let's try it for a few months, 6 months, a year. You asked about time scale. I don't think anybody really knows. But let's do another test. Do we see improvement? We're working on research right now to try to tease out some of these things in the context of some clinical trials we've been running where, we've done physical activity interventions or diet interventions or things like this. We know that the participants in these trials lost weight. So in terms of the phenotype we're interested in, we know that there was an improvement, and now we're doing blood assessments of them sort of pre and post to figure out, is this being tracked in the biological age or the proteomic age signatures as well? So I guess the answer is stay tuned. From our research, we don't know. I would say in general, in the field, I don't think we have a great sense in terms of the granularity of at what time scale does a DNA methylation clock or a proteomic age clock or a transcriptomic clock, at what time scale do they react meaningfully to some change in your environment or your behavior or your diet or something like that. I would also say that it's not well known or cataloged which are the things that will influence those. There are 50 diet interventions or way more than that. You can assess, which are the ones that are going to have a meaningful change. That's one of the other things we're looking at now. We're trying to use some of these large human population data sets to try to start to catalog what are all the things in your environment you can be exposed to, like pollution, housing, proximity to noise, all these things, diets, lifestyle, behavior, all these things. What's the kind of catalog of all the things that are going to track with these biological aging profiles? Because that's ultimately what matters, and what makes these enticing is that it's able to tell you about some kind of future risk early enough. It gives you a window of opportunity to change something. So we, as the research community, need to be able to compile some kind of evidence that gives us an idea of, these are the most strategic things you could do to have the biggest impact. For me, the whole idea is strategy and strategic. I think that's why aging in general is a strategic approach, because it tells you about biology common to all these diseases, and gives you, the ability to kind of triage not just one test per disease, but one test for many diseases. So to sort of strategically start from there, figure out where you need to go, and then if we can complement that with what are the highest priority interventions or things you can change to move the needle in the best way, then I think that will have just a huge impact on being able to change the way we do preventative health.
So you mentioned earlier, so you've got thousands of proteins in this data set, but you can do a pretty damn good, you build a model that's almost as good as the one that uses the full data set using something like 20 proteins. Yeah. I want you to talk a little bit about some of those 20 proteins and what they are. I think one thing that could be useful for people is, are any of these things that are in a lot of standard blood tests? You mentioned, for example, C-reactive protein. A lot of people that get blood work done. I've got blood work data of myself that has CRP in it. Are there any common ones that people might actually have some data for themselves on that were, part of that core 20 that were really informative?
Well, like I mentioned before, the list of 20, the set of 20 are fairly multidimensional. So you have a number of proteins that have different functions and are involved in different biological processes. I'll name a couple of them. So for example, like I said, you have proteins that are involved in extracellular matrix dynamics. The most important protein in the whole model is a protein called elastin, which does kind of what it sounds like it should. It's sort of involved in elastic fibers in your body. It's part of the sort of cell structure and things like that. Also in that category are collagens. So another of the top 20 proteins is a collagen.
I see. And so I assume these things would basically just index the quality and youthfulness of one's skin.
Well, they're also associated with cardiovascular aging. I mean, think about like one of the things that happens to arteries and blood vessels as you age is they stiffen, right? So these are really important, especially for cardiovascular aging, but they've been implicated in other types of, aging-related diseases as well.
Basically the quality of your connective tissue.
Exactly. So those are some of the most important proteins, and elastin is the number one most important protein in the model that we developed. Other types of proteins that are important are involved in inflammation and immunity. So you have things like chemokines and cytokines, not C-reactive protein. C-reactive protein is an interesting example because C-reactive protein is another one of these things where even though it's a biological signal, is associated really consistently with different health outcomes, cardiovascular disease, things like that. But again, going back to our old trick of Mendelian randomization and using the genetics to infer something about causality, the sort of consensus now is that C-reactive protein doesn't actually have a causal impact on a number of the diseases we use, but we use it just broadly as a marker of kind of systemic inflammation or something like that. So C-reactive protein does not come up in this panel of the top 20, but maybe in light of what we now know about whether or not it has a causal role in some of these phenotypes, might not be surprising that it's not there. We also have different proteins involved in development, in metabolism, and we have a number of neurodegeneration-related proteins. The two big ones that come out are called neurofilament light, usually abbreviated as NEFL, and GFAP. Interestingly, for your listeners who are familiar with the kind of genetics of Alzheimer's and dementia, they'll know that one of, or the strongest sort of genetic determinant of Alzheimer's and dementia risk is ApoE4 genotype. We have ApoE protein in the panel that we assessed, and it does not come out in our model. So there is some kind of interesting disconnect, again, between the genetics of what we know and the actual protein level. So again, speaks to the importance of pulling out the proteins. I don't know enough about, and we weren't able to get to the bottom of why it was that ApoE protein wasn't coming out in our model, whereas the ApoE genotype is so important for discriminating risk for Alzheimer's and dementia. But that's something to come out. But in general, if you think about the only two sort of reliable and reproducible genetic signals for longevity, one is ApoE, and the other is a protein called FOXO3. Both of those proteins were part of this 3,000 protein panel that we started with. Neither of them end up getting selected by our model. So, we keep coming across these interesting disconnects between the genetics of longevity and aging, between the sort of genetic regulation of longevity and aging through DNA methylation or transcriptomics or things like that, that we see played out in the proteomics, but ultimately it's the proteome that's going to be driving these phenotypes. So it's interesting to look at from that perspective.
You looked at some associations between aging and various biological, physical, and cognitive functions. What were some of the things that were most closely associated with faster aging or just aging generally?
Well, it's hard to answer that question because, to be truthful, almost everything we looked at was associated. And now we did that in a sort of intelligent way where we only started by looking at things that were sort of known to be age associated. So we didn't look at every phenotype under the sun, but we tried to build as comprehensive a list as possible of all the functional, cognitive, biological, and disease phenotypes that have some suspected or known role or a consequence of aging, and then look at whether this biological aging signature is associated with them.
As a way to basically validate your model.
Exactly. Yeah. And to understand if it's capturing aging broadly or is it picking up only certain aging of certain organ systems versus others. What we ultimately end up finding is that it seems to be capturing aging at a systemic level that's broadly and across a number of different organ systems, which you see play out in the different types of diseases that are associated. So we have liver diseases, kidney diseases, heart diseases, lung diseases, brain diseases, cancers. But then in terms of the physical and biological phenotypes that you mentioned, you've got cognitive function, lung function, BMI, blood pressure, hand grip strength, walking pace, things like that are measures of kind of like, again, physical frailty and functioning. So in terms of which were and which weren't, the sort of short answer is most of what we studied was associated, and that's one of the things that was so remarkable, that we weren't expecting it to be so broadly associated with everything. But then we can start to ask on top of that, what was the biggest effect? Which that does start to discriminate some of these. Again, if we think about the age-related diseases that we tested, by far the biggest effect that we see from the proteomic aging signature is on dementia and Alzheimer's risk. In terms of the physical phenotypes, I can't remember pull up the image of the figure off the top of my head, but there were a number of kidney and liver markers from the blood biochemistry panels that were particularly strongly associated with very large effect sizes. We did a number of things to try to make sure we tested that robustly. We adjusted those for all kinds of measures of kidney function, liver function, eGFR, things like that. And it was very robust to those kinds of adjustments. And again, I'll also say as an aside, that's one of the benefits of, again, being able to do this in these kinds of cohort studies, not just that we can do things on a big scale, but we can, because we have the power in terms of numbers of samples and number of people who have diseases, we can adjust really robustly in our models for things. So this is another thing that you'll see more often that you would like. And again, coming back to your question of how do we pull out a high quality study versus one where we don't necessarily trust the results. If I see a paper that shows some miraculous association, but then I look at the model and they've only adjusted for age and sex, I don't believe it. Show me that association persists after we adjust for education, income, race, ethnicity differences in where you live around the world or in your country, until we adjust for your smoking level, your physical activity level. I mean, we even adjusted for people's BMI and hypertension status in these models. And, we have one figure where we show just as you go increasing level of covariates and adjustments you put in these models, the effects just stay the same. So again, for us, that's a signal of robustness.
Yeah.
This isn't just some result of we did one model and it's really brittle and it's really precarious, and if we just add one more covariate, the whole thing goes away. And we find this even though we saw really large effects for these liver and kidney markers, things like creatinine, cystatin C. After putting in a whole barrage of adjustments for these liver and kidney function markers, it's still there. So for the physical measurements, liver kidney function, frailty index was another really strong association in terms of the size of the effect. But in terms of the diseases, dementia and Alzheimer's really stand out as by far the biggest effect size.
Longevity's obviously become a very hot topic, and that's great. People should care about this. We're learning more all the time. There's a huge commercial component to this as well. There's all kinds of people, pushing supplements and protocols and this, that, and the other. How much do we really know about aging, and how much can you really push it if you focus on this? So, for example, I think his name is Brian Johnson. The guy I'm talking about. Yeah, he's one of these longevity gurus. This guy's as extreme as you could get. He's putting millions of dollars a year into his own personal health. I think he's getting blood transfusions from like his 18-year-old son or something like that. He's taking a very particular regimen of supplements and foods and everything, and he's trying as hard as anyone could try to enhance his longevity, enhance his health span. Let's say you take someone like that and you compare it to someone who's, we'll just call run-of-the-mill health conscious person. They're in the gym 3 or 4 days a week, but they're not an Olympic athlete. They're mindful of their diet, but they're not very totally neurotic about it. They're pretty damn healthy. They make informed decisions, but they're not spending millions of dollars a year to go to extremes. What's the delta between those two people actually likely to be? In other words, how many extra years do you think someone like a Brian Johnson, someone going to those lengths, is actually buying versus someone who's just sort of generically health conscious?
The simple answer is we have no idea. I think that particularly when you get into supplements, longevity drugs, longevity treatments, all the evidence is in mice and in animals. There's no longevity human trial because it's too expensive and too long to do one.
Yeah.
So already the level of evidence that we have, I would say, is uncertain, the effect to which it will work and be effective in humans, and what that delta will be that you're describing. How many years of life are we really buying in humans? I just don't think we know. And, even in terms of lifestyle modifications, all of the extreme things he's doing, I don't think, like I said, I don't think we have a great catalog of, again, the things we know that work robustly in humans across populations. We just don't have a good catalog yet. We know for specific age-related diseases what are the effective things. So again, what are the effective things for preventing the major killers, cardiovascular diseases, heart disease, dementia, things like that. Physical activity, lifestyle, don't smoke is the biggest possible thing you could do. But beyond that, I would just not be able to put a number on it for you whatsoever. And the big unknown with what someone like Brian Johnson is doing, put aside for a moment whether the degree to which we're certain with any individual protocol he's using or implementing works in humans and has some appreciable difference, put that aside for a second. We are in especially unknown territory. What happens when you put them all together?
Right.
And that becomes particularly important for supplements and pharmaceuticals and drugs and things like that. There's this idea of polypharmacy where we might know how one thing works if you just do that one thing, but then things will start to interact and work together. And so that's, in my mind would be a complete unknown. And I'm not saying that to necessarily disparage anything that he's doing. I'm just, I couldn't tell you with any certainty.
Yep. There is, but this is an important point because there is a very, very important implicit assumption that people like that are making, which is certainly it's true that you can be deficient in a nutrient and that, supplementing with the nutrient you're deficient in is probably going to be a good thing. But to assume that, more of all of those things simultaneously is good, that's a complete X factor. We just don't know.
Yeah, I think it needs to be tested. And there are two things that make me uncomfortable about this particular approach. One is that it becomes a real moral gray area to make recommendations to the public to do things. If we aren't sure as a scientific community, we have the right level of evidence that it's safe and that it works. But number two, I feel strongly that being able to live well and age well should not be something that's only accessible to someone who can throw millions of dollars at it per year. Our thinking is that, aging should be a really strategic way that we can try to develop tools and approaches that democratize living well and democratize good health and preventative health that can have an enormous economic benefit because we can start to be strategic and lower cost. So I'm much more interested in developing low cost preventative screening tools and low cost, interventions that A, we know work for as many people as possible, and then B, can be used and accessed by as many people as possible. I think that's going to really move the needle on, being able to curb rising rates of multimorbidity per year, which is just the number of people who have two or more diseases simultaneously, have a huge impact on lessening the burden of chronic diseases on the healthcare system, which has obviously a personnel toll, an economic toll, an emotional societal toll. So these are all things that I think are more important. And I think I just don't know if I am behind the message that the only way to make an impact on this is if you're a millionaire. I think there must be things that we can do and that we can create that can democratize and not just make this another axis along which we're going to create more health inequalities on top of what we already have. And so, that's what I think is more interesting in this whole space, not who are the few elite people who are going to live to be 200, but how can we develop something and learn something that's going to let everybody live, add more years of, healthy disease-free life to their lifespans, regardless of whether we're all hitting 100 or 120 or something like that.
And obviously it's possible to live to be relatively healthy into your 100s. Like people have done it. It happens. Even super centenarians are out there. Do you think, can you sort of comment on, the idea that, we could get to a place where, the average human being can expect to be a centenarian, super centenarian versus, can we push the human lifespan? Like, is there a cap on the human lifespan that's going to be sort of a fundamental limit here? Is it even possible under ideal conditions, whatever that means, to live to be 150, 200 years old?
Again, I'll just put my hands up a little bit and say, I have no idea. And I don't think that the scientific community has a good idea. This is very hotly debated. Is there a genetic limit on lifespan? We don't know. Human lifespan in the last 200 years or sort of human life expectancy in the last 200 years has roughly doubled from 40 to 80. And
how much of that, do you know how much life expectancy has gone up if we sort of subtract out infant mortality and things like that?
Well, that's all part of it. I mean, part of the reason why it has human life expectancy, at least in some countries in the world, has increased so rapidly is from improvements in sanitation, public health, development of antibiotics, effective healthcare, effective treatments, effective medical systems. So it's part and parcel, I think, what's happening more broadly. I couldn't tell you a specific number. If you take out infant mortality, how much does it change? But certainly on the aggregate, on the population level, that's definitely what we see. And we certainly do see now versus earlier that there's, again, one of these sort of J-shaped, where there's a sort of a higher mortality risk in general, at least in the UK population, which is where I've looked at these data most closely. In the first years of life up until, early teenagehood, there's sort of this increased mortality risk that then decreases. And then after that, there's an inflection point, and your mortality risk just starts to go up with age again after that. So even in the present day, there is still a greater risk of mortality in early years compared to when you're a teenagehood, where there's this nadir, and then you just start to increase steadily as time goes on and the decades go on. But what it all points to is that all of these changes and all these increases in life expectancy have happened way too fast for genetics to be the major culprit. It's been changes in our environment, changes in our healthcare systems, changes in the medicines and the treatments that are available to us. And that plays out generally when we try to look at the genetics of aging and longevity and lifespan. You see, the heritability, or sort of the amount of variability in those longevity or aging phenotypes that are explained by genetics, or at least, genetic polymorphisms, to be in the 10 to 25% range. So the thinking is that a lot of what's going to make the difference for you is not, there are some people that are more or less lucky in terms of genetics, but there's a lot you can do in terms of how you live your life. Whether there's a cap, like we can't live past 150 or we can't live past 200, I just don't know. And I've never seen anyone give convincing evidence that they know the answer to that. But I think everyone would be curious to know.
So what are some of the things that you're working on now or working on next with respect to some of this proteomics work?
So we're working on, I guess, three major things right now. I've touched on a few of them already. One is we're also looking at the genetics of these proteomic aging signatures that we've developed to try to understand to what extent is variability in proteomic aging rates explained by genetics, which are the specific genetic variants, are those genetic variants that are similar in other kinds of phenotypes that are related to aging. So trying to build out our understanding of the biology of looking at these biological age signatures by using the genetics to be able to compare then to all the genetic research that's happened for so many other diseases and phenotypes. Mm-hmm. So that's one area that we're looking at that we're very interested in. The other area, which is the opposite, is we're trying to, like I mentioned before, go deeper into the environment. So across biobanks and in the context of some intervention studies, tried to catalog what are the things that you do, what are the things you're exposed to that are going to associate meaningfully with these levels of biological aging. Mm-hmm. Is it pollution? Is it diet? Is it physical activity? And we're trying to, with a lot of that kind of work, we are trying to do it in the same spirit as we would do with the omics or all of the biology, even though it's environmental influences, trying to put them all together in measures of what we call the exposome, something which is sort of just, if you think of the genome being all of the genes, the exposome is kind of all of the exposures that you might be exposed to across the life course. And you get that same benefit that I mentioned earlier, where that helps us find what are the signals across everything you're exposed to that are the biggest signals, taking into account the rest of the environment. So again, this idea of being able to prioritize and strategize, if we could look at all the environmental factors, which is the one that's going to get the best bang for your buck, which is the one where by far it's going to be the biggest impact. So we're interested in tackling it that way. We have another paper that's under review looking at that now. And then the third axis is, again, as a continuation of this work, is to try now to figure out and understand how we can use these kinds of proteomic aging tools and models in the context of intervention trials and clinical trials and randomized controlled trials. So can you use this as a surrogate biomarker in a clinical trial, where you don't need to wait 20 years for someone to develop heart disease. We can do an assessment in a year and see if there's been a significant biological improvement or sort of biological decline. So that's one area to see if we can use it as a readout for whether an intervention was successful or not, where we can compare it against, people actually lost weight, or they, did or did not develop a disease we're interested in. So these are the three areas where we're trying to look in now to figure out more about the biology of proteomic aging, to figure out more about what are the things we can do that can change it, and what are the applications we can have for it that will either improve our ability to figure out interventions that will improve Healthspan, that will improve people's risk of disease and the ability for people to live more and more years without developing these major chronic diseases.
Mm-hmm. For the commercial clocks that are out there, people can buy something, send in a sample, and get their biological age compared to their calendar age. Are those at least reliable? Are they going to be directionally accurate? Do you think those are worth looking at? Do they actually work as advertised, basically?
The epigenetic, I'm only aware of commercially available tests that are based on DNA methylation. There are some based on glycans and different sort of protein modifications. But the major offering would be DNA methylation clocks. At least on the population scale, they accurately capture age. And on the population scale, they have been associated in sort of one-off studies with different diseases of interest, depending on which one you use. There have been some interesting papers that came out. I'm thinking of one from Morgan Levine's group, for example, where they showed that if you do sort of a duplicate sample where you take the same person and you take two blood samples and you run the methylation clock on the same person from the same blood sample taken at the same time, you don't get the same result. And they have developed new methods using principal components analysis and other statistical analysis to try to basically pull out that noise and get something that's more reliable in terms of like within the same person across samples, so that ideally, if I took 10 blood samples from you right now, you should get 10 measurements of the exact same biological age readout. So I think there's dangers with things like that. I think, there's many individual papers on these epigenetic clocks. There aren't any huge population scale papers, just one paper looking at large populations and certainly replicating across tons of different populations. There's lots of different individual studies. So I think it will definitely tell you something about age, but I think we know a little bit less about the phenotypes that connects with, if that makes sense. And I think, what we need to move a lot of these tools forward is, first of all, really good human evidence that will tell us in a rigorous way, not just does this capture your age well, but does this tell you something meaningful about risk for the conditions that we care about. But I can't really comment too much about the specific tests that are available out there. I'm not too familiar with all of the different analytics they give you, but at least on the population scale, they should be able to capture your age very accurately. But whether or not, at least on the population scale, that methylation is really causally linked to the change in biology that's going to be the driver of aging-related phenotypes, I think that's a little bit of an open question for me based on what I've seen.
Is there anything that you want to reiterate from our conversation today, or any final thoughts you want to leave people with about aging and longevity, broadly speaking?
I guess I'll say one thing about aging and longevity. Again, my interest in this field is not to discover something that will make everybody live to be 200-300 years old. As you mentioned before, I think there's a lot of hype and excitement around longevity medicine. There are longevity clinics popping up. There are tons of people who will tell you about your biological age or sell you a supplement that they think will help you live longer. But I suppose I would reiterate that the amount we know on any of that for certain is not as high as you might think. But more broadly, I think that there's a more interesting way that we can think about aging and longevity research, which is thinking about it as a way that we can totally revolutionize preventative healthcare. Again, if aging biology is something that's common to all of the major age-related diseases, so all of the major chronic diseases, and by the way, if you look in the global population above 70, 9 out of the top 10 causes of death, they're all chronic diseases. They're all age-related chronic diseases. So there's a real strategic opportunity to focus on aging, which is going to tell you something about the biology that's common and shared across all of the major killers in the global population. So if we can develop tools and approaches based on that, it's going to have a huge impact on our ability to prevent and to develop strategies and treatments and interventions that will help us prevent a number of these major killers simultaneously. So I would be happy if life expectancy stayed the same, but we totally moved the needle on Healthspan, which is how many years are you living disease-free? That would be a complete win to me. And I think aging research and studying aging biology is something that can really help us get there in a really strategic way. I think there have been some interesting economic estimates of the value of slowing aging in the US population, at least, 1 year versus 10 years. The number they put on that is in the trillions of dollars. If you can truly do something to stall aging by using the biology that's common to all these major diseases to do something to just prevent the onset of these diseases. If you prevent one by using aging biology, you're probably going to have a knock-on effect to prevent many other. And so, that's our hope for the aging field. Our hope is that we'll continue to do this research in humans in large data sets across different diverse populations, where we can make sure that we find signals that are really stable across people, and that aren't just noise. I mean, that's our hope where the field will go, and that's my hope for what we could use for this field and what we could use this research for most effectively.
All right, Austin, we covered a lot today. Thank you. This is interesting work. I look forward to seeing what you're going to come out with in the near future. Thank you for your time, and I hope to talk to you again at some point.
Thanks so much. It was great to talk to you.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.